Welcome, everyone. Thank you for joining us for today's track on the epigenome in health and disease. It's my pleasure to introduce to you Dr. Michael Snyder, who is the Stanford Ascherman Professor and Chair of Genetics and the Director of the Center for Genomics and Personalized Medicine at Stanford University School of Medicine. Dr. Snyder is a leader in the field of functional genomics and proteomics and one of the major participants of the ENCODE project. His laboratory study was the first to perform a large-scale functional genomics project in any organism and has developed many technologies in genomics and proteomics. They have been used for characterizing genomes, proteomes, and regulatory networks. Dr. Snyder is the author of the book Genomics and Personalized Medicine. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Dr. Snyder's keynote presentation is titled Big Data and Health. My name is Judy O'Rourke, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone this event is interactive. We encourage you to participate by submitting as many questions as you want during the presentation. Simply click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box. Our speaker will respond to your questions via email following the presentation. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're experiencing a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Now, without further ado, please join me in welcoming Dr. Snyder. I will now turn the presentation over to him.
Okay, well, thanks very much for having me here. It's really a pleasure to present our work to you. I do have a number of conflicts. They're all listed at the bottom of the slide. So I'm going to start with a premise that many of you may not like to hear, but it's really that the way we do medicine these days is all wrong. This is, in fact, a slide from Stephen Hubble it's the next slide that presents that. But this is where the way we do medicine is on the left, which is that we're presently very focused on treating people when they're ill. As such, it's very reactive. We measure very, very few things. The frequency we measure people when they're healthy is usually rather rare. And in general, nearly all decisions based on medicine are based on population-based measurements. In contrast, I think most of us would agree we should really be focused on health. As such, be very proactive. We're capable of measuring many, many more things than typically measured in a doctor's office. The frequency, I will argue, should depend on what you're at risk for and your health trajectories for those particular areas that you are highest risk. And lastly, I think all of us would agree that medicine should be based on the individual, not based on population-based measurements. And so we should really be focused on precision health rather than precision medicine. And so to illustrate this point, this is a slide I've gotten from Stephen Hubble, which is it's probably many of you think that the standard temperature when you take your oral temperature is 98.6. That's what we're normally taught. It turns out, though, if you actually do a study like the one shown here, there's several of them out there like this. This one actually measured 3,000 people. It turns out, first of all, that number is wrong. The median number is not 98.6, but in fact, 97.5 is probably a lot more accurate. And not only that, if you look at this box at the bottom of the box, that's the 25th quartile, and the number is 94.6, whereas at the 75th quartile, that's the top of the box, that's 99.1. So the point is that there's actually a spread, and this is of people's healthy baseline state. And so what it means is that people have different oral temperatures. And so if your normal oral temperature is 94.6, and you go to a doctor today, you walk in the office and you come in at 98.6, they'll tell you you're perfectly healthy, everything's fine, you're in great shape, what are you doing here? Go home. When in contrast, you're really up 4 degrees over your baseline measurement, you're probably rather sick. And you would never capture that because you didn't know the baseline measurement. So the point is these population-based measure decisions are not always in your best interests. So we think, again, this theme I'll present many times, we think it's very, very important that you understand what your baseline values are, and then you can find deviations for that to better capture your illness. So the health state, if you will, depends upon many things. Depends upon your DNA, as I'm sure most of you appreciate, but it depends upon other things such as exposure, shown in the bottom left, as well as lifestyle, your exercise, the food you eat, the various activities you do, all contribute to your health state. And I think I envision a world where in the not so distant future, people will be born with their genome already sequenced, and then you'd like to understand what kinds of activities, what kinds of things you do will influence your health in some probabilistic fashion. That is to say, if you're at risk for Parkinson's, you may not want to be a pesticide worker because that greatly increases your chances of getting Parkinson's. So we think it's very, very important that you have the relevant information available so that you can adjust your lifestyle accordingly. So as many of you probably appreciate, there's now a revolution in being able to collect data that we think will impact your health. The one that gets the most attention is the top one shown here, which is genome sequencing. You can now get your genome sequenced for $1,000 or less. The interpretation we will argue is costs much more, at least to do it very thoroughly the way we do it. But nonetheless, you can get your genome sequenced for under $1,000, and in the future, that number will become much cheaper even. We would argue there's been an equally strong revolution going on in mass spectrometry, so you can now measure many thousands of proteins and metabolites in bodily fluids like blood and urine. And so you're capable of getting a much, much better molecular picture on people using these tools. And more recently, we would argue there's been a revolution in the wearable technology, so you can actually get continuous measurements off of people. And so in fact, we use all of these different technologies and others to be able to better capture information that we think is relevant for people's health. So as many of you may know, a number of years ago, we set up something we call personal omics profiling, where in its most deluxe forms, we measure people's genomes. We do that once, but then we measure other omes, and in its most deluxe form, we measure about 13 other omics measurements. A typical scenario, though, is probably measuring 8 other omes on people, and we follow them over time. So this is a project started about 9 years ago, a little over 9 years ago now, actually studying me. And now we've been running it for over 6 years on 107 other people and collecting just lots and lots of data. And specifically, if you go down from the genome, you'll see epigenome. That means a DNA methylome we measure, transcriptome. Most of this is off of blood components, specifically peripheral blood monocyte cells. These are your immune cells. So we'll measure the transcriptome, proteome. We measure off those cells in plasma. This is with something called TMT labeling and mass spectrometry. We also have immunoassays for following 63 cytokines and growth factors. For the last 5 or 6 years now, we've been optimizing both metabolomics and lipidomics to be able to get much, much deeper profiles there, autoantibodies. And we've also been doing mostly 2 different microbiomes, gut and nasal, that we collect samples for measuring others. This is on top of making very deep clinical testing. We have questionnaires, and we have a lot of glucose measurements you'll see in a bit, because our cohort, it turns out, is enriched for people at risk for type 2 diabetes. About half of them are either pre-diabetic or diabetic. And then we also have, if you look over to the left, we have wearables. We started that about 6 years ago as well. But so in general, what we do is we sample people quarterly, so every 3 months or so when they're healthy. And then if an adverse event comes along, like a viral infection, we'll take many more samples, typically about 5 more samples. And then other events as well, though. We will actually measure people, for example, if they have a colonoscopy, we'll measure them before and after. And we have a lot of different kinds of food perturbations, other sorts of studies running as well. So again, we've been running this, the slide's a little bit out of date, but it's for 6 years for most people, some a little bit more, some a little bit less. And so we're collecting all kinds of deep data. So why are we doing this? Well, we're really trying to understand what it means to be healthy. Most of these individuals are healthy, say a fair number of pre-diabetic, which is typical of a lot of the US, by the way. But we're trying to understand what it means to be healthy. How does that differ amongst people? How does it change over time? And what happens when people undergo some of these perturbations, like a viral infection and such? What does that mean to their health? What exactly is going on? So as you might imagine, when you carry out a study like this, you collect really an extraordinary amount of data. It's unbelievable. And by the way, most of the data is actually publicly available. Not all, but the vast majority of folks have agreed to make their data, including their genome sequences, open access, so all of you can get access to it. It's at the microbiome website, if you will. It's called the Data Coordinating Center, where all those data reside. And so we've collected a ton of data, and we actually have all kinds of very interesting stories around this. But what I'll tell you over the next 30, 40 minutes or so are some of the vignettes of interesting findings we have. So it turns out from the genome alone, we find quite a few interesting results. So just from the first 70 people, we had 12 individuals who have clinically actionable information. That is to say, they have mutations in their DNA they should know about that will actually affect their health care. And an obvious one that many of you are probably familiar with is this one at the bottom of the list called BRCA. We had one individual with a BRCA1 mutation, which puts that individual at higher risk for breast cancer. There are others here as well. If you go up at number 4 from the list, that RBM20, one individual has an this RBM20 mutation, pretty young person, and it turns out that predicts a mutation and it leads to cardiomyopathy. And sure enough, it turns out this individual has cardiomyopathy. It was picked up in a subsequent stress echo after getting the genome sequence. And so it's pretty clear, in fact, they're on drugs now. So it's very clear that in this individual, finding this mutation was very, very important for their health. It turns out their father and aunt each had heart attacks. Father actually died fairly young of his heart attack. So the point is that these can be very, very important pieces of information to know. Just to give you another example, if you look third one down, HNF1 alpha, that actually is in an individual. 9 of our folks are actually thought to be type 2 diabetic when they enrolled. It turns out this individual who was classified as type 2 diabetic was not type 2 at all. They're a different kind of diabetics called MODY, and they've been on the wrong medication for years. So they're actually shifting the medication as a consequence of this information. Because the way you treat a MODY person, very different from a type 2 diabetic. At the very top of the list, we had 1 and 2 individuals, that's what 2X means, who had HSPD mutations, and one of the individuals had a whole body MRI and turns out had early thyroid cancer, which can be associated with this mutations in this gene. And so actually she had that part removed and was able to then maintain the other half of her thyroid, which then she doesn't need to go on thyroid replacement hormone. So the bottom line is many people had useful information from their genome sequence alone. It's not the majority, it's 12 out of 70, but if you're one of those 12, it's a pretty big deal. Now, more recently, we've come up with new ways of analyzing genomes. That's shown here, where we've come up with, in this case, for abdominal aortic aneurysm. So generally, when people analyze complex disease, what they do is they try to find all these common SNPs and add them up. These days, people add up millions of SNPs to try and predict disease risk. And that works for people at the far end of the spectrum. The problem is it's very hard to work. It may not work so well at other parts because we think that genetics is not very linear. You can't just add up things. 1 + 1 doesn't always equal 2 in the case of genetics. If they're in the same pathway, 1 + 1 equals 1. And if they're synergistic, 1 + 1 can equal 4. So the point is you can't just add up genetic variants and expect phenotypes to come out in a linear fashion. So we actually decided to take a different tack for looking at complex disease, which is looking at rare variants, which is not normally done. It's normally just done for Mendelian disease. We decided to try this for a particular disease, abdominal aortic aneurysm, which is kind of a big deal. It affects, if you're 65 years or older, about 10% of people actually get this, and typically will die. You'll see in a minute. And it's even overall in the population, it's relatively prevalent. The problem with it is that with abdominal aortic aneurysm, the way it's usually discovered is people's aorta grows and then it bursts and they die. And that's typically how you're found to have this disease. So it has a 90% mortality rate. So obviously getting an early predictive test would be a very, very big deal. So the new approach we used was, rather than look at common variants, we said, can we look at rare variants and set up predictive tests from this? And so the genetic underpinnings of this disease are not very well known at all. Just a few genes have been implicated. It really isn't known what the genetic architecture is. And so we set out, we sequenced 401 genomes, excuse me, to high quality. 268 are cases, if you look at the left, and 133 are controls. So these are whole genome sequences, and we sequenced to a depth of about 60x, if you look at the top. We then called, rather than looking for common variants, we looked for pathogenic and likely pathogenic variants. We called that throughout the genome. And then we did a machine learning algorithm with a 10-fold cross-validation to look for sets of genes. We didn't say how many, we just said, give us sets of genes that would be hypermutated in the cases relative to controls. And even though it's only 401 genes, with this approach, the machine learning actually picked out 60 genes that were hyper-enriched in cases relative to controls in this 10-fold with a very, very strong p-value, 10^-13. And so it's a very, very robust set of genes that seem to be enriched in cases relative to controls. And then we went on and validated this by many means. We went out and got the mouse orthologs of these, and sure enough, they tend to be enriched in heart issues. We actually could assemble modules around these genes. So these are the interacting partners of these 60 genes, if you will. And then looking at the pathways involved, it's all the things you would expect: blood circulation, blood pressure, cardiomyopathy, aneurysms, another module, and so on. So these are exactly the sorts of genes. They're misregulated both in humans that have AA as well as in mouse models of AA. So again, it seems to be the sorts of genes you would expect to be involved in this disease. Finally, we could go on and actually set up a predictive test for the disease that's shown here, where from the genome alone, you do okay. Just from a genome sequence, that's this red line on the right, you can actually make a predictive test of about 0.7, so it's not perfect, but it's okay. Just from a genome sequence alone, predicting risk for disease from these 60 genes. If you add in a little bit of EHR data, basically what you'll find is, sorry, you can see they have light savings things here at Stanford, so went in the dark a moment there. Anyway, if you take the genome sequence, add a little bit of other clinical information, you will in fact improve the prediction quite a bit. You'll get it up to about 0.8. And it looks modest on the EHR, adding that over EHR plus genome looks modest over EHR alone, but it actually greatly decreases the false negative rate quite a bit. So the bottom line out of all this is that with the genome sequence, we can now set up a predictive test for disease that was normally discovered when someone actually dies. And so to see how it compares with other tests, we show this here. So these are all the clinical tests that we had out there, could find. And basically from the genome alone, it's okay. It's that blue square there. This is a predictive value for specificity and sensitivity. But if you look at it from the genome plus EHR, it's a pretty decent test. And it's as good as, or better than many other tests that are out there now. And what's nice about it is it's actionable information. If you're at high risk, you should actually elevate your HDL, and that will dramatically lower your risk. We can show that in other studies I won't show you. So the bottom line out of all this is, by this new genome analysis approach, we can find ways in which to predict people at risk for a complex and fatal disease that didn't exist before. And we do it now. So it's some predictive value right from birth, but even better after a few clinical measurements. So that's just one of the kinds of stories we've been getting from this study. Overall, it turns out by profiling this group of 107 people this is from about the first 4 years of data, we had 49 major health discoveries. So by doing big data analysis, we had 49 major health discoveries that you found with this approach that you would not find by normal visiting the doctor's office kinds of things. These are again, things that came out of our big data analysis. So I mentioned 12 from the genome, but there are lots of other things as well. And I'm going to show you a few examples in a minute. So lymphoma, we found one case of early lymphoma. That's in the bottom area. So the major categories we've lumped them into are metabolic, cardiovascular, heme-oncology, infectious, and a variety of others. So as I say, in the heme-onc, we had one person with early lymphoma, 2 people with pre-cancer, one called MGUS, I'll show you that in a minute, another was smoldering myeloma. Finding these is a big deal because you'll keep an eye on them. We had, in addition to one person with a heart defect I mentioned earlier, there's a second person who had a significant heart issue. Turned out that was due to sleep apnea, so that we believe is now under control. And there's a variety of other things as well. And I'll walk you through a few examples right now. So this next slide, this shows an example of someone with a early lymphoma. This was picked up by imaging. So by actually doing ultrasound, they were doing, half of our group did a stress echo. This individual did as well. And they actually they imaged the spleen at the same time with ultrasound. And one person turned out had an enlarged spleen, and they had other elevated markers in their blood as well. And sure enough, it turns out they had early lymphoma, which was caught. What was kind of interesting is in retrospect, we could find a marker that made a lot of sense. It's this graph at the bottom. The peak is actually when we detected their enlarged spleen, and then they were treated, and they underwent remission right away. You can see that in the lines that went down. But there's a very interesting marker shown here that actually tracks with the onset of this disease and makes sense. So we have a possible new marker for lymphoma that didn't exist before. Another example is shown on this next slide. This is an individual that actually was found from outlier analysis. So there's 1,000 data points here. One individual who had been sampled twice at that point, his 2 data points, also a fairly young person, had way too many IgM cells over what you would expect. So everybody else is in that bunch down at the bottom. And this individual just has a ton more IgM cells and more IgM than anyone else in the bunch. So it's pretty, and then went on and had a formal diagnosis of MGUS. And since 15% of MGUS folks wind up going on and getting aggressive cancer, finding this out now is a big deal because you could actually then monitor them more closely and catch it if they ever do turn to aggressive cancer. There might even be things you can do that you help reduce your chances or better treatment by preparing for it now than waiting until you get it. We had 6 people who actually had plaques in their arteries. I'm one of the 6. And as this individual here, you can see where the red arrow is. There's just a little bit of plaque in the artery. In all 6 cases, wasn't known. That was true for me. And in all 6, they had a recommendation of increased statins. That was true for me. And my plaque is now actually shrinking a little bit as a consequence of doing that. As I mentioned before, there's a lot of interest in type 2 diabetes for this study. That's simply because I'm actually type 2 diabetic, and actually in my case, it was predicted from my genome, which I then did get. I'll show you some data on that in a minute. But we were enrolling people who were either at risk. A lot of them were just plain old normal folks we enriched as well. And going into the study, on the left bar is what people thought they had. There were 9 individuals that thought they were type 2 diabetic, a few that thought they were type pre-diabetic. That's that orangish bar. And it turns out at study enrollment, 2 people are diabetic who didn't even know it. So we discovered that right away. A ton of people are pre-diabetic and didn't know it. Turns out 9 out of 10 pre-diabetics don't know they're pre-diabetic. And then during the course of the study, others either had diabetic values or were formally classified as diabetic. 21 other people had diabetic range measurements. 9 individuals became formally diagnosed with diabetes, and a number of them went on metformin. One of them actually lost weight, which helped. So what was interesting to us was, how do people become diabetic? Do they gradually work their way in? Do they spike in? What are some of the underlying ways in which people get diagnosed? And what are some of the underlying mechanisms by which people get diabetic? Well, it turns out people get diabetic in different ways, and that's shown here. This top one, that's actually me. That's about 7 or 8 years of data actually there. Then there's 2 other people. As I say, we had 9 people who became diabetic. two people spiked their way into diabetic, and I'm one of those two. So if you look here, the orange line is fasting glucose at the top left panel. Orange line is fasting glucose. The gray line is hemoglobin A1C. And I actually was going along just fine. I first spiked, if you look at the left side, into diabetic range measurements after a nasty viral infection. That's actually not a mini spike. That's 8 months of data, from 8 or 10. And I went up by both fasting glucose, hemoglobin A1C, and OGTT. I got it under control initially with lifestyle, but it did spike back again later. And that time, I never ever really got it down. It kept drifting up, got higher. It's a little complicated. I stopped running, which is about the time it first spiked up. Oh, sorry, the second jump up there. And then when it got pretty high, then I discovered it was high again, and I started running, got it somewhat lower. But anyway, I'm back to type 2 diabetic. There's one other person that spiked their way into diabetic range. Most people just gradually became diabetic. So they slowly became type 2 diabetic over time, and they got there by different measurements. The one at the bottom got measured by something called oral glucose tolerance test. The one in the middle by fasting glucose, others by hemoglobin A1C. So it does seem like there's heterogeneity in the way people are both diagnosed and the way they became diabetic. Two individuals, by the way, seem to associate became diabetic in conjunction with a large weight gain. And then the others actually became diabetic without a weight increase. So most people became diabetic without gaining weight, in fact. And so again, the underlying causes are very, very heterogeneous. And then it turns out the way they are diabetic is also very, very different. If you can see here, we measured rates of insulin secretion and a ton of other measurements on people, insulin resistance and so on and so forth. And you can see different people. The top guys are people who secrete insulin normally, pretty quickly after taking glucose. Others are more and more delayed. The ones at the bottom are quite delayed. And what we found is there's just enormous heterogeneity in people, in the way people are diabetic and the way they have glucose dysregulation. And I'm a good case of that. I am not a normal type 2 diabetic. Most type 2 diabetics actually insulin sensitive. In my case, most type 2 diabetics are insulin resistant. I actually happen to be insulin sensitive. I make insulin just fine. I'm actually a slow releaser. I'm one of the people at the bottom. And so, in fact, the way I should be treated for type 2 diabetes is very different than the average person. I don't respond to metformin. I probably should take increased secretagogues, which actually promote secretion. So the point out of all this is that there's many, many ways in which people can have glucose dysregulation, and understanding the underlying mechanisms is going to be very, very valuable, we think, for treating them. Now, another area that we've looked at is, in fact, DNA methylation. So many of you may appreciate that DNA methylation is associated with lifestyle and other sorts of things. And so it's thought that your nutrition, exercise, aging, the environment can all affect DNA methylation. And this is associated, by the way, with epigenetics. Now, DNA methylation is associated with gene silencing. So if you're methylated, your genes are methylated in the promoter, in general, they're usually shut off or reduced in expression. And so there's a lot of interest in trying to follow DNA methylation because it can be potentially, it's another way in which you have gene regulation, and it's one affected by ways in which you have some control, again, your food and exercise. Although how this whole thing works, I should emphasize, is not really understood at all. And it is also interesting, your methylome changes with age. So we actually decided to go and look at, for me and a few others, what happens with methylation over time. And so we actually, in my case, measured my methylation throughout the genome using something called whole genome bisulfite sequencing. So we can measure all of my methylated bases, if you will, they're called Cs, CpGs in particular, tend to be methylated. And we did this over the first 3+ years of my measurements that were taken. Now, during this time, first 3 years I was in the study, maybe 3¼ years, I had been through 6 viral infections. They're all shown here. They're all common colds. Yellow is rhinovirus, very common. Red is respiratory syncytial virus. That's a rare case. And the 2 blue ones towards the bottom are adenovirus infection. Again, all very common colds. And during this time, I had taken 57 samplings, if you will. So we decided we would follow both my RNA, my transcriptome, as well as my DNA methylome across this time period. And so we did the transcriptome at all 57 time points. For the methylome, we did 28, and we did less because it's a very, very expensive experiment, actually, do the whole genome by sulfate sequence. We did 28. So we did the viral infections, and then we did several of the healthy time points. We didn't do them all, again, for cost. And what we discovered is, this first part's not a surprise. What we discovered was that the transcriptome changed a lot every time I had a viral infection. So during each of these 6 viral infections, if you look at the left panel, each of those colored boxes is a viral infection period, and it starts at the beginning of the period. And so those top genes of the bunch, each row is a different gene, each column's a different time point. And what you'll see is that there were many, many changes during each of these viral infections. That's not a surprise. If you look at the genes that change, you can look at the categories that's shown on the right. It's your immune response genes that are changing every time you get a viral infection. Again, that's not a big surprise. What was really cool was the next slide shown here was, even though my RNA was changing every viral infection, my DNA methylome only changed twice in the exact same population of cells that were measured. And the 2 times they changed are the 2 times when my glucose went off. So if you see those little dots, it's probably hard to see. They're at the end of the red box, and there's another set that starts appearing at the end of the last yellow box. It's at the top of the left panel. If you look at those little dots there, sorry, I can't point this out to you, but that's when my glucose is elevated. It's the same plot I showed you earlier. And what you'll see is that my DNA methylome actually changes. Each row is a region of the genome that's showing changes, and this happens to be regions and promoters. And what you'll see is that there are 2, if you will, reddish regions. Now, these aren't huge changes. There are several percent, but they do go off, and it typically precedes when the glucose peaks, actually. So we think these are early events. It starts about 60 to 90 days before the glucose peaks or hits its peak values. And again, it happens each time a glucose went off. And furthermore, if you actually look at the types of genes where these promoters are sitting next to, it turns out it's your metabolic genes that are actually showing these differential regulations. So if you do Go analysis, your metabolic genes are actually showing differential DNA methylation during these times when your glucose is dysregulating. And in fact, many of the genes are, in fact, involved in glucose methylation. So what we think is happening is that your methylome is a measure, if you will, of chronic disease, whereas your transcriptome is a measure of acute disease. And that makes a lot of sense. The methylation changes are probably not quite as dynamic and maybe more permanent in nature, in the sense that if you think about aging as a permanent kind of response, your methylome changes with age. So again, we think your methylation is measuring chronic disease situations as opposed to acute disease. Yeah. Another very important finding of this study is that everyone is different. So no matter what ohm we use, people will cluster by themselves, not by a condition. And I'll just show you, these are the healthy time points, but it'd be true even if I sprinkled in the disease cycle. Now, to keep it simple, this is 12 people with at least 10 healthy baseline visits, so a lot of visits over several years. I happen to be the one in light blue, so I'm a part of this study. And you'll see that each color is a sampling. Each dot is a different sampling, and each color is from a different person. And so people will cluster these healthy visits, like this is 6 years of data for me in this light blue, and they all cluster according to me. And same is true for the brown, the reddish. Each color, again, is a different person. So if you look at clinical labs, you've got cytokines, metabolite, not so much for the transcriptome stuff. For all the other realms, people very, very strongly cluster by themselves. And so what that means is that your personal signatures are really quite robust. That is to say, you're very different than the person sitting next to you. And in fact, that turns out to be, for the most part, greater than even if you get sick or something else. That is to say, people with viral illnesses, or we did a weight gain, weight loss study, you will shift if you gain weight, but you will still look more like you than anyone else. So your personal signatures, we think, are really, really robust. In fact, if you were to get off your seat at the end of this presentation and someone took a swab, in principle, if they had the right reference, they could tell it was you who sat there, because you'd be leaving behind a very robust signature. So again, we argue that these personal signatures, and to me, this is the real reason why you need to do personalized medicine, personalized health in individual level. You have to know people's baselines so you can see the difference between the person when they're ill relative to healthy. It's hard to tell someone who's getting ill relative to everyone else healthy measurements. You have to know your difference relative to your healthy baseline. Okay. So the other area that we've been spending a lot of time on lately is with this wearable technology. So this really, I'd say, came on a number of years ago, mostly just for activity tracking, fitness, things like that. And we wanted to see, starting about 6 years ago, whether we could use these as useful health measurements as well. And so these days, believe it or not, there are 900 of these portable sensors on the market, smartwatches, et cetera. And so we went out, we've tested now about 30 or 40 of these over the years. The big issue is getting access to the data. So we actually, as I say, tested many of them. At the end of the day, we settled on 8 or 9 of them I wear every day. In fact, maybe you can't see this, but I'm wearing 3 smartwatches. This ring is not really a ring, it's a sensor. So there are these portable devices that let you take all kinds of measurements. And as I say, I wear 8 or 9. The rest of the cohort usually uses an app on their smartphone, along with a smartwatch that we picked out for them. So again, these devices, they make all kinds of measurements. They'll measure your activity, they'll measure your heart rate, skin temperature, sleep. They're devices for measuring blood oxygen, even radiation monitors, and of course, steps, et cetera. So they measure many, many different things. And what's powerful about these devices is they measure them continuously. So a typical smartwatch will make about 250,000 measurements a day. The Lux ones will make many, many more. So we're getting a lot of data in real time. So we've used these for tracking all kinds of things. You can set up and figure out people's personal baseline parameters, for example, their circadian patterns about heart rate, skin temperature, and we collect all that data. And then what you try and do is actually find deviations from people's regular patterns. And I'll just give you a few vignettes here. One of the biggest deviations for me from my baseline is actually when I fly. It turns out this is known already in literature. Your blood oxygen will drop in airplanes. It's known. It's actually not well known. Most flight attendants don't know it, and a lot of pilots don't know it. But basically, on the left is a panel that shows a flight from San Francisco to San Diego. Green is altitude, so the plane goes up and comes down. And blue is my blood oxygen. So it starts out around the space, I think it's about 97, drops down to lower for the short flight, 93, comes back up when you land. And that's true on pretty much every single flight I take. It's not only true for me, we've now measured somewhere around 80 different people. Everybody, with one exception, everybody's blood oxygen drops on aircraft. Okay. It turns out, I won't show the data for this, but it correlates with fatigue. So in fact, we think the reason you're getting tired on aircrafts is because they drop the air pressure in the plane and your blood oxygen drops and you get tired. And for some people, it drops a lot more than for others. In my case, about 5% of the time, it'll be 90 or below. And then most of the time, it runs around 93. Some people would even be lower. What was powerful is that we actually found these kinds of measurements are very useful for figuring out when people get ill. And I actually figured out my Lyme disease from a pulse ox and from my smartwatch. And so the backstory behind this, before I dig too much in data, the backstory behind this is I was helping my brother put up fences in rural Massachusetts. And then 2 weeks later, I was flying to Norway through Frankfurt. And so on this last flight from Frankfurt to Oslo, which is a pretty low aircraft, your blood oxygen does not normally drop too much on those. And green, so the left-hand panel in green is what my blood oxygen normally does on a short flight of that sort. And blue is what happened on this particular flight. My median value, if you look in the middle, at cruising was about 90, which is pretty low for median value. Turns out, and then when I landed, if you look at the left side of that panel, turns out when I landed, my blood oxygen stayed at 95 and then come back up to normal, which is close to 100%. So the bottom line out of all this is that my blood oxygen is abnormally low. I looked at my heart rate. It's not shown here at high resolution, but if you look at the right panel, this is daily averages, if you will, outline measurements to be precise. Then what we noticed was at the time, my heart rate was up. Later, I learned my skin temperature was off too. So the bottom line out of this is that my heart rate, skin temperature, and blood oxygen were off. And it turns out that right after that, I got a low-grade fever. I did, after several days, went to a doctor in Norway, and I warned him ahead of time, it might be Lyme disease, because the timing of 2 weeks seemed a little suspicious. And I did not get a bullseye, by the way. And he actually pulled blood there in Norway, and you know, looked at my monocytes, said they were up, and said, "Yep, you've got a bacterial infection. You should take penicillin." And I actually said, "No, I think I should take doxycycline." As you might imagine, it got tense for a few moments there. He did give in, gave me a 2-week supply of doxycycline, which I took, and it cleared it right up. And so within 24 hours, I was right back to normal. And when I got back, that's that positive sign on the right panel. I actually drew blood and was measuring. Sure enough, I was Lyme positive. And it turns out I'd given blood before I left for Norway, serendipitously. So, and it turns out I was Lyme negative. We tested that blood then. So the point is I had seroconverted during that time. And the net reason, the bottom line is that I was able to figure out my Lyme disease by an abnormal drop in blood oxygen and elevated heart rate. So we could detect a pretty serious disease just from these simple devices. Well, that prompted us then to go look in detail at all of the data I had at that time. So I had 2 years of data from a smartwatch. And we looked at, if you look at the left, we look at, it works best for outline measurements, things true standard deviations or more. The skin temperature on the left, and bottom is, sorry, it's a little messed up there, but that's actually outline heart rate. And it turned out over these 2 years, there were 4 times when I had high CRP and was probably ill. 3 times I reported being symptomatic. And so there's a Lyme disease case, that's the triangle days. These are daily averages, if you will. There were 2 viral disease, the top one and actually that bottom high CRP2. And there was a 4th time where I didn't report being ill, but I had high CRP, same levels as being ill, probably successfully fought off. And the cool result was that every single time at elevated CRP, at high outlying heart rate and skin temperature, every single time. So that says that, in fact, yes, you probably can pick this up with a smartwatch. So we wrote an algorithm, we call it change of heart, that detects the earliest times when you get ill by elevated heart rate over your resting background. And it works really quite well on my 4 times I get ill. We tested it on 3 other people, one of whom got ill twice. And every single time, we can see this change in heart rate over resting background well before people reported being symptomatic. So we think that this is an early measure of illness. Again, occurs before you're symptomatic. The worst case is going to show up at the time you're starting to feel a little bit congested, but we actually think it shows up before that. So we're now rolling this out as a 1,000-person study. You can roll if you have a smartwatch. I should point out skin temperature didn't work very well for us, I think, because not everybody wears their watch tight enough. But heart rate works very, very well. So that's what we're testing. And so the goal will be to tell people when they're getting sick before they realize it. Okay. We think that has a lot of value. Another area that turns out to be very, very impactful is for wearables is glucose dysregulation. So I mentioned before that 9 out of 10 pre-diabetics have no idea they're pre-diabetic. Well, these days are these simple glucose monitors you can wear. Right now you require a physician, but in Europe and probably at some point in the US, you'll be able to get them without a physician's prescription. So we put these on normal and pre-diabetic on a few diabetic folks to see what their spiking patterns look like. And it turns out we saw different kinds of patterns as shown here. So the top line shows an individual with good glucose control, doesn't spike much in 24 hours. Next guy spikes some. Bottom one person spiking a lot into pretty high levels. So, excuse me, we've basically written algorithms to be able to more systematically classify people. And we classify them into what we call glucotypes. So the idea, I won't get into details here, but we can go through and actually pick out, basically, we go into these glucose measurements and we pick out the patterns of something called spectral clustering. And then we can classify the patterns of the percent of time people are in these patterns. And each row here is a different person on the right side. So each row is a different person. The ones at the bottom, these are people with a low spiking glucotype. The ones in the middle are the moderate spikers. The ones at the top are severe spikers. I'm a severe spiker, by the way, because I spike a lot. So we've also put people on standardized meals. I won't have time to go into this, but some people will spike more on bread and peanut butter, others on protein bars, and so on and so forth. People will spike to different foods. And Aaron Siegel's lab has nicely shown the microbiome is an important part of that. One of the interesting observations we have is that nearly everybody spikes the cornflakes in milk. So this is like poison, we would argue. It's probably worse than alcohol, the way everybody's spiking cornflakes in milk. We think that's not so good for you. So we can also then look at, again, I mentioned we put these on normal people, pre-diabetics, and so on. So the left-hand panel just shows classifying people by their spiking pattern. So this pinkish red means you're a severe spiker, green a moderate spiker, blue means you're have good glucose control, don't spike much at all. And what we can find is that people, we've classified all these people by fasting glucose, hemoglobin A1C, OGTT, but we find people like these two with the arrows pointing at them that are severe spikers, but by all normal diabetic measurements, they actually look normal. They don't show any sign of fasting glucose problems, hemoglobin A1C, or OGTT problems, but yet they're severe spikers are spiking just as much as diabetics. So we think there are people running around there with some pretty severe glucose dysregulation who have no idea that, and would even get missed by common assays. So we think these devices will ultimately be very, very powerful for diagnosing glucose dysregulation. So the last story I want to tell you is an area that I feel like we've never been able to get at very well. So we've spent a lot of time on this. This is your exposures. So we're very interested in trying to understand what is your exposure at a personal level. And we decided, just for starters, to work on airborne exposures, because we all breathe and we suck in things all the time. And I guarantee probably none of you right now know what you're exactly breathing at this moment. And yet it might be good for you, it might be bad for you, you just have no idea. So we think this is a big deal because obviously biotic exposure, meaning pollen, other things, is very important for allergies and asthma. Abiotics like chemicals, it's very important for toxicity and cancer and so on. So we think it's very, very important to measure all these things. And if you think about it, a lot of the basics about the human exposome just is not known. So for example, we don't know how big is the exposome. I mean, it's usually exposed to 10 species, 1,000 species, 100,000, who knows? And chemicals, same things. How dynamic is the exposome? Or we're always getting exposed pretty much the same thing? Or does it vary a lot from place to place? And or is it more seasonal effects? Who knows where the different exposures are coming from? And how does the chemical and biological exposure relate and so on? So the bottom line was that we basically set up a method for following personal exposure. So we actually took an air monitor, it's sitting in my backpack right next to me. It's a small device that sucks up air, and we modified it to actually it has a pump that sucks in about 1/15 of what you breathe. And under the intake, we actually have a particulate filter. So we measure, it captures all the particulates, biologicals, and inert things. And then underneath that, we have a chemical absorbent, a cartridge with chemical absorbent. And so for the biologicals, what we do is we'll take the filter and we actually harvest DNA and RNA, and we sequence incredibly deeply. And for the chemicals, we actually take the chemical absorbent, elute off the chemicals, and do mass spectrometry and measure as many chemicals as possible. And so we're using very high level technologies to see what's in these samples. And then we follow people over time. And so in a typical scenario, I'll wear the device for typically one device during the week, one on the weekend. And every time I go to a new location, Boston, China, whatever, I'll take a different device and make separate measurements. So we can follow location, we can follow, weekly versus weekend measurements, this sort of thing. And we've now profiled, something got lost here, I hadn't noticed this before, but basically all the color is evaporated here. Sorry about that. But the bottom line is that you can't see this, but I'm P1. Imagine this is going to be off on all the slides. I didn't see it before. But P1 is me, and I've been wearing the device for now, it'll be 6 years this summer. We've collected 2 years of data that we've analyzed initially in this study. P2 is actually worn the device for a year, P3 a little over 3 months, and then we have up to 17 other people worn them in different circumstances. I'll explain some of that in a minute. And you can't see on the right side, but I travel all over the world. So we have tons of measurements from all over the planet, so we can see what the exposure looks like, different locations, also different seasons, so on. It turns out P3 travels all over the US quite a bit, so we have some there. P2 not so much traveling, but all right. So bottom line is we can make all these measurements. And so we've sequenced a ridiculously deep amount of reads. In total, we've done over 70 billion reads, 10 trillion base pairs, and looked at these results. And about 40 billion of the reads are from DNA, 30 from RNA. So it's quite deep. And so in total, we've measured over 2,500 species. There's typically about 300 species per sampling. Even the person wearing the exposometer for 3 months had over 1,000 species. So conclusion number one is that the exposome is vast. You will get exposed to many different species just by walking around, breathing, and so on. Okay. We did a little bit of validation with scanning. How dynamic is it? Well, this is me, P1, P2 is supposed for a year, P3 for 3 months. And basically, each color represents a different kingdom. So red is bacteria, this brownish, orangish thing is fungi, green, of course, is plants, and blue are animals. And so what you'll notice, just look at me on the timeline at the top, as you look at across time, the exposome is quite dynamic. It depends, from week to week, location to location. We'll get into some more detail. Same is true for P2, same as for P3. Bottom, we've clustered these. You can see the left side is mostly plant exposure days, then there's bacterial exposure. Most are actually fungal exposures. The last part's mixed, and it's true for me, P2, P3, so on. So the bottom line is we will have different exposures in different sampling periods. So then the next question is, what is this due to? Is it due to season? Is it due to location? What's driving your dynamic exposome? And the answer is it's due to all of that, both locations and seasons. And so the left just shows my samples and sort of a PCA kind of plot, if you will. You'll notice that the Asian samples are the reddish ones, is on the left side. They're in that reddish circle, so they're more on one type of a sample, and then other locations or others. And the same is true for season. We can see some effects there. And we can quantify this. This is what's kind of cool. So for a biological exposure, we can quantify how much we can assign. We can't assign most of it, by the way. We're still trying to figure out where it's coming from. My guess is very, very specific location. But the bottom line is we can say for what we can assign, a lot is due to location. But it turns out environmental, which is mostly season, actually is another big player. And some of it's due to technical artifacts. So With that in mind, we actually did a whole bunch of follow-up studies to follow location. I'll just show you one of these. Oh, good, this should show up. So this is actually a very defined experiment to test the effect of locations. We followed 4 people over the exact same month with these exposometers. And the one in purple lives in San Francisco. The red one here, that light blue person lives and works across the bay. And someone who lives pretty close to me and works close to me is the green one there. And so my samplings, because they travel, are pretty scattered. Same with the light blue, they're a little more scattered, they travel. You'll notice the San Francisco ones are pretty tight, and the Sunnyvale ones are pretty tight as well. What's special about this is that everyone has their own exposome, even if they live pretty close to one another. As I say, location is very, very local. It's very area specific. So I'm very different from the Sunnyvale person, who's very different from San Fran, and so on and so forth. So we're all very different in our location exposures, even if we live close to one another. What's kind of cool, let's see if that shows here yet. It's great, is that you can actually then see what are the species that are driving us. And there's a number of species at each of these locations. But I thought some fun examples out of the San Francisco exposure is that actually you pick up sludge bacteria is one of the things that tends to be more specific for the San Francisco exposure relative to the others. And if you've ever walked down Market Street in San Francisco, you can probably understand why this result happened. So anyway, the point is you can map out specific organisms with specific locations. We do pick up sequences very similar to pathogens. I'll come back to this in a minute. These are very, very low level in the samples. We also have strong seasonal components. There's hundreds of species that show seasonal effects. So the green ones are the spring abundant organisms, the light blue or summer, the red or fall, and the darkish blue or winter. So again, there are hundreds. Many of these are known already. A lot are new. 2 examples are shown over on the right. You'll see the pine action pieces known already, late spring, early summer. This turkey tail mushroom is more like late fall, early winter, and so on and so forth. And you can actually correlate these to allergies. Really a lot of fun. I mentioned that you actually can find, they're very, very rare. Sequences very similar to pathogens. Again, very, very uncommon, but they are in our samples, and so we're pursuing this further. We do find seasonal effects of chemicals. This is the chemical results. We find all kinds of chemicals in the samples. There are plastic components in every sample, pesticides, even some carcinogens. Their abundance depends on location, very, very influenced by location. And you can even pick up, this is a case of some things that correlate with rain. You probably know you can smell when it's going to rain. It's because the air pressure drops and this compound geosmin comes out of the ground, and you actually smell it, and that's how you know it's going to rain. It turns out when it rains, plastics, I'm happy to say, wash into the ground, so you're not breathing them as much. Maybe that's good, maybe it's bad, maybe you're drinking them. Body scent comes off you when the air pressure drops. Pesticides come out of the ground. Maybe that's not so exciting. So these are the things you can learn from these things. One of the coolest results we have is shown here. This is, in fact, we wanted to correlate biologicals and chemicals. And what we found is this nasty chemical at the bottom called pyridine. It's often found in paints and other things. It inversely correlates with fungi. So wherever you have pyridine, which is, if you look over at that light blue one in Boston, there's a lot of pyridine there in that sample. And they're low fungi. And you see it's a perfect anti-correlation. So what we think then is that pyridine is probably inhibiting fungal growth. And so if you're allergic to molds, this actually might be useful. Maybe you want your house painted with a pyridine paint, keep the fungal molds down. Now, I don't know, maybe you'll die of cancer, but at least you won't get black mold allergies if you probably have pyridine in your paint. So this is kind of stuff you can decide on your own, I suppose, what you want to use. The summary of all this is that we can actually build personal exposure clouds. So this is mine. Everybody, not surprisingly, has a different cloud. A lot of human-associated sequences in here. That's not surprising. A lot of this reads, I didn't say are human. We've subtracted those out from 70 billion. But you can tell what people have. I have cat, dog, and a guinea pig. They all show up in the sampling. We get the plant and environmental and insect exposures and so on and so forth. So yeah, these are, in fact, what you can do, and you can build people's personal clouds. What I think is going to be powerful about all this is that all of these data you can collect in your smartphone. And now we're building a personal health dashboard, so we can actually start setting up exactly what we can stream the data in. You can see it live for your wearables. Now we're getting microbiome and other data, so you can see exactly how your health information is tracking, and see it in real time, and then follow you over previous times and see what's going on. And share this with your physician so they can better understand what's going on with you. And lastly, this is how I see this whole thing rolling out in the future, that people will, this is the Mike Snyder world. I see people getting their genome sequenced before birth, together with other omics and wearable measurements. You can actually follow people more carefully to see what they're at risk for when early signs of disease, and be able to better intervene. So lastly, I do want to credit, I have an amazing lab. These are just some of the folks who have worked in this project over the years, Wen Yu Xiao, Brian Penny, Now Going, John Paul, Reza Solani. I see I've got to update this slide, and others have all worked on this. Verbals, Shao, Jesse, Dennis, Sophia have all worked on this. George Weinstock has been a fantastic collaborator. His group has helped with microbiome analysis. Trace McLaughlin on the clinical side. Exposure work is done by his talent group in the bottom right with Chao, Shin, Laura, Singh, and Shan. And we've been supported by the NIH, and that's been terrific for making this project possible. So thanks again very much for having me here. If you have any questions, I'm happy to answer them online. Thanks again.
Thank you, Dr. Snyder, for that informative presentation. I would also like to thank LabRoots for making today's educational webcast possible. As a final reminder, our speaker will follow up with any questions you've submitted via email. That's all for now. Thank you for joining us. We'll see you next time. Goodbye.